Okay. Yeah, so thanks very much. So today what I'd like to talk about is some work we've been doing over the last few years to try to better understand some of the molecular and gene regulatory changes that happen in the human brain during development. And this is actually a project that we didn't really intend to get into, but somewhat were led to through other projects that we're attempting to do. So about a decade ago, and now we had a study where we're focusing on DNA methylation and looking at the changes that happened to DNA methylation patterns and states during human brain development from gestation through to adulthood. And what we found is that in contrast, if we look at the levels of methylation in the genome, in contrast to the canonical form of DNA methylation in the CG sequence context, what we found is that there was this non-canonical form of DNA methylation, CH methylation, that is associated with gene repression, and its levels really skyrocketed up postnatally, particularly coincident with the process of synaptogenesis. So we thought this is pretty interesting. This is this distinct form of methylation. It seems to be linked to gene repression. It's highly specific to neurons, so we'd like to study it better and in more depth and to try to identify mechanistically whether it had a role in gene regulation. So when I started my lab back in Australia, what we wanted to do was to generate in vitro models of this system to try to functionally perturb it much more easily in allowing screening and so forth than compared to in the brain. But whenever we generated neuronal cultures, be that 2D cultures or 3D organoids, and looked at the level of this type of methylation that accumulates postnatally, as you can see in the normal brain, what we found is that we essentially saw none of it. So the human neurons that we would generate in vitro, essentially at this level, were resembling fetal neurons. And from our work and a range of other work, looking at different molecular layers within the cell, it was also clear from transcriptome, from proteome, that the cells that were generating in vitro, the neurons were resembling a fetal state, which has been shown by a number of studies and is actually well known to occur in a very broad range on most of the cells that are generated from induced pluripotent stem cells or embryonic stem cells. So they have this sort of arrest. So he said, okay, well, we can't do this initial screen and I guess mechanistic analysis of the CH methylation, but maybe then this is an important barrier that we'd like to overcome to be able to get mature neurons in culture, because presumably they're going to be more effective and useful for modeling postnatal disorders and for studying neuronal function as maturation processes have proceeded. So we turned then to the broadly used approaches of trying to force transcriptional changes or cell identity changes through manipulating transcription factors. So for example, say neural progenitor cells, and then activate transcription factors that may drive maturation. But then the challenge was, which transcription factors do we choose to do this? So we began to look for data sets that were out there of transcriptional changes, so we could predict transcription factors or identify transcription factors that seem to be associated with the maturation process in the human brain. But what we found is that the studies were limited to bulk tissue analysis, so bulk brain tissue, which really is confounded and challenged by the underlying complexity of these tissues and the many different cell types. And as an example, if you were to look at trying to overcome this through single cell or single nucleus analyses and plot the number of studies or samples that have been studied from the prefrontal cortex of humans by age, we noticed that there was this real gap in paucity of samples and analyses that had been done, particularly from third trimester through to teenage and young adulthood. So clearly, this is a critical period in which lots of developmental processes have happen. Of course, in the early stages of development, the first 2 trimesters, there are key processes that are going on within brain development of gliogenesis, synaptogenesis, and neurogenesis. But in this long, protracted process of development from largely postnatally up to adulthood, there are similarly key developmental processes that occur and cellular processes that occur, which are important to understand of how the adult human brain is formed. Not only are there these cellular and developmental and structural changes, but there are really key developmental and cognitive milestones. And if you imagine a baby versus a toddler or a child or a teenager versus an adult, they're vastly different in their behavior and capabilities, whether it's communication or their social or emotional characteristics or their cognitive characteristics or capabilities. And these occur at predictable timeframes during development. So clearly there must be gene regulatory processes driving cellular and connectivity processes that are occurring during this period that are driving the increased maturation and complexity and capabilities of the brain. Yet I found it quite surprising that we still, years after the technologies existed to do this, we still have very little idea of the transcriptional changes that might be underpinning some of these processes to form essentially the characteristics that define being human. So then what we somewhat got distracted and said, okay, well, this looks then like a gap that would be valuable to be filled. And we set about getting samples from the human prefrontal cortex to try to chart from gestation to adulthood the transcriptional or the gene expression patterns and regulatory processes that occur during this period. We, from the NIH Neurobiobank, obtained a pretty wide range of samples from the human prefrontal cortex of cryopreserved tissues and performed single nucleus RNA-seq to map gene expression states and single nucleus attack-seq on many of these samples to map regions of chromatin accessibility, which can give us insights into transcription factors that might be driving these transcriptional changes. Here's just shown a plot of the different stages that we studied from the gestational age 22 weeks through to the oldest was a 40-year-old sample. And the circles just indicate the assay, attack-seq or RNA-seq that was done and the ages of them. But ideally, what we were aiming to do was to fill as best we could this gap in profiling that had been undertaken. So we set about doing this, and first I'd like to talk about the transcriptome profiling that we did. So we performed single nucleus RNA-seq on all of these samples and generated about 150,000 high quality nuclei transcriptome profiles from this. And first encountered a range of computational challenges, actually, and analysis challenges, which we had to overcome to integrate the data. So we generated this data, we're able to integrate it using a new approach of downsampling. And what we could see after we do this, and this is just a UMAP of all the cells and their transcriptome, is that if we color all of the cells by the age of the sample that they came from, you can see this nice progression from fetal stages through to adulthood. And you get this gradient of changes, which indicates ongoing transcriptional changes through development. Another way to look at this is just to show the cells that come from each of the individual samples progressing chronologically, where you can see that in these different cell types, these inhibitory neurons, principal neurons or excitatory neurons, oligodendrocytes and astrocytes, you can see this temporal progression through there, indicating that there are ongoing transcriptional changes that take place through development. So to define a bit more structure in the data, we first started by defining subclusters of the data, and then to identify what cell types based on marker analysis these clusters came from. For example, layer 2, 3, CUX2, principal neurons, or VIP interneurons. What we could then do is take all of the cells that belonged to a particular cell type, say layer 2, 3, CUX2 neurons, or earlier progenitors of those cells, or early immature forms of those cells, and we could group them together into what we call a trajectory. And a trajectory represents all of the cells of a particular cell type over the whole developmental span that we were looking at. We focused on 14 major trajectories in principal neurons or inhibitory neurons, astrocytes, oligodendrocytes, and so on. Underlying those were more complex trajectories, 45 sub-trajectories that end in many of the different subtypes of the different neurons or non-neurons, but for many analyses, we just focused on the 14 major trajectories. And the first thing we want to do is to use these to allow us to map differential expression. What are the gene expression changes that are happening over development? And so we call these developmentally differentially expressed genes, or DevDegs. First, what we could do is to take a very high-level view of when are most of the transcriptional changes taking place. So we could take these DevDegs, genes that are changing in abundance over development, in each of the different cell types shown here in different color, and we could look at some very broad characteristics of them. So we fit a trend to each of the genes, to expression levels, and first looked at a couple of measures: the rate of change of expression or the acceleration of expression change. And both of these, we see that many of the changes, transcriptional changes, or most of them occur at very early stages of development, particularly around birth. So rapid rate of changes of expression from prenatal stages through to 1 year old, and then fewer in childhood, but there are ongoing changes. Or acceleration of the change in expression, which peaks at birth, which indicates that the rate of these expression changes is coinciding with this really major event. What we could do, having these differentially expressed genes as a side note, is also to identify better markers of maturity, where really we've been lacking in good markers that indicate true postnatal maturity or the drop in immaturity, which allow us to study this through other means. So, for example, we could find good markers of immaturity, which taper off in adulthood, or really good markers of maturity, like RGAP26 or KCNH1, which in a large fraction of cells accumulate to high levels over development, which are useful tools for further analysis of maturation through independent means. For example, shown here through RNA scope detection. Another aspect that we're interested in is looking at all these different cell types that we find in the prefrontal cortex and asking, well, do they all mature at the same rate? So do we see over all these different cell types that are reaching an adult state of maturity at the same rate? Or is there actually different rates at which different cell types reach maturity? So we analyzed this timing of maturation. We did it by focusing first on all the adult cells, and we know they are adult cells because they came from an adult sample. And then we could for each cell type, look at the cells along the developmental trajectory from each sample, and ask for all the cells for each sample, how many of them have nearest neighbors that are adults, or adult nuclei, basically. So we're starting to look at the cumulative proportion of those. So we're starting to count as development proceeds, how many of the nuclei that we're profiling of a certain type start to look mature, look like an adult nucleus from the transcriptional perspective. And we can do that for each of the different trajectories. Indeed, we did this for all of the sub-trajectories as well, 45 of them. What we see is that there is this, there's a range of different maturation timings. We see, for example, if we look at the principal neurons, excitatory neurons, the characteristic inside-out organization of maturation, where we see the layer 5, 6, appearing to mature earlier than the layer 4, layer 2, 3. And we see various other timings for a range of other different cell types. But it's more complex than this. If you, for example, just focus on these principal neurons, we can cluster these different types of neurons by their maturation timing and see that actually there are a range of different cell types that appear to group together with the more similar times of maturation that may have some relevance. So we see what we refer to as waves of maturation that seem to start in infancy or in childhood or in adolescence, and cell types that are starting to mature at these points that may be related to formation of early sensory related circuits in infancy or in childhood, in the formation of intracortical to interregional connections, or in adolescence, the process of cortical myelination. But this just gives us some new insights into the differential rate of maturation, how these change over this protracted process. Of course, since we had all these differentially expressed genes, what we could start to do is to look for patterns of them. What were the different patterns of expression changes that we see over development? Which cell types did they occur in, and which processes were they associated with? So we clustered all the developmental differentially expressed genes into what we call gene trends, which just are groups of genes that in particular cell types, which could be many cell types, show a similar change in transcript abundance over development. So you could have genes that are rapidly upregulated early in development, or more slowly upregulated through development, or genes that are transiently upregulated but then downregulated, or downregulated then upregulated, or downregulated. And not surprisingly, given the number of genes and the complexity of the process, we see many genes in each of these different categories showing these diverse dynamics that occur over development. And we can look at which cell types they tend to occur in, their abundance, and different processes that we're associated with. As I won't go into detail in this, but we can look at in a much closer level and focus on particular groups of cell types. So this was taking a focus on interneurons, and in the interneurons we see 26 distinct subtypes that we could identify. And using the data, what we're able to do is to visualize and predict the developmental trajectories of these different cell types over development. So we could use approaches such as SC velocity vector analysis to predict which cells related to one another. And we can constrain that by the chronological knowledge we have of the data, since we know what the ages of the cells from the samples they come from, which limits these predictions and allows us to refine somewhat these trajectories and developmental processes. And through this, we're able to identify and resolve the developmental origins of particular subtypes of inhibitory neurons from either MGE or the CGE. Of course, we have all these changes in expression that we can profile and analyze, but what we're particularly interested in, because we ultimately want to be able to drive maturation, is what are the regulatory processes that control these expression dynamics. So if you recall, I said that in addition to the single nucleus RNA-seq, we also perform single nucleus attack-seq. So this is mapping of regions of accessible chromatin throughout the genome at single cell resolution. And so these accessible chromatin regions are often indicative of regions of the genome that are bound by transcription factors, for example. So you can find association between accessible regions in expression and try to predict the transcription factors that might be driving the expression changes. So we mapped chromatin accessibility over many of these samples, over 17 of the samples, almost all of them overlapping with samples that we did the single nucleus RNA-seq from. And using ArchR, assembled this data again into a chronological map of chromatin accessibility in different cell types that takes place over development. And again, we can see, if we look at the age of these nuclei, we can see a chronological ordering particularly within the neurons from fetal stages through to adult stages. Again, indicating, unsurprisingly, really, since we see transcriptional changes, that there are regulatory changes as well that are taking place through this process over years and decades. Now, this on the right is just an example of a snapshot of a region in the genome where you can see clear regions of accessibility over all the different stages of development. We group samples into these 6 stages for different cell types, where you can see the different cell types show clearly different patterns of accessibility or change in accessibility over development at different regulatory regions. What we want to do is identify transcription factors that might be driving the expression changes. So we use SERAT to integrate the RNA and attack data, matching the nearest neighbors to each other. And what we're able to do then is to analyze the association between cell type specific chromatin accessibility within 250 kb of transcriptional start site of a differentially expressed gene and the transcript abundance of that gene. So we could look for a correlation between an increase in accessibility of a local, within 250 kb, a local region of the genome, and expression of that gene. And through this association, infer that region showing this change in chromatin accessibility may be a regulatory element, so a putative cis-regulatory element that is bound by a transcription factor and driving a change in gene expression of that gene. So we do this. Having different cell types and groups of pseudobulk groups of cells allows a prediction then of this association between accessibility and expression. We predict about 300,000 cis-regulatory elements that are linked to genes, and most of them are distal, so they're not close to the gene at the transcriptional start site. One thing we can do for a quick sanity check of that is to take all these CRE, so cis-regulatory element gene links. We take all the cis-regulatory elements and cluster all of the cells based on the accessibility, so the attack-seq signal within the accessible region. And so we have these clusters of attack-seq data. And then for each of them, in a parallel plot, show the transcript abundance, the gene expression patterns for those. And we see this nice correspondence between the expression of the RNA and accessible chromatin. So it's just, in a way, indicating the correspondence between these two signals and the likely good, high-quality prediction of these links between chromatin accessibility and expression. Using this, then we can start to take groups of genes that show similar dynamics over development. So in these gene trends, so coordinated changes in expression over development, and we can search within them for enrichment of particular transcription factor motifs within the cis regulatory elements that are linked to those genes that are changing that particular way of development. So, for example, upregulated genes in this gene trend G4, what are the transcription factors, or in this case, transcription factor families that are enriched in the cis regulatory elements linked to those? And we find a whole these are transcription factor families. There are many different transcription factors underlying this, but we find transcription factor motifs that are enriched for all these different gene trends and that may contribute to driving their expression changes. An example would be down-regulated genes. We see are enriched in developmental transcription factor motifs, which makes sense. And a number of these we can use attack footprinting indeed to see that we get a depletion in the attack signal right at those sites, which indicates likely a protein binding at these sites. I won't go into detail here, but you can choose any sets of genes that you're interested in then to predict transcription factors that might be driving the changes in expression of those genes. For example, dynamic ion transporters that change in particular cell type in a particular way over development, and then predict the transcription factors that might be driving that. We weren't only interested in process of normal development, but also interested in how these processes might be disrupted, leading to neurological psychiatric disorders. And of course, we know that various neurological psychiatric disorders have very specific and reproducible ages of onset and of progression. So what we want to do is to explore how the transcriptional dynamics over development might link to genes associated with these disorders. So what we could do is to take a range of different neurological disorders or psychiatric disorders and take the genes that have previously been associated with these diseases. So we've got these sets of disease-associated genes. Of those genes for each disorder, we can take the subset of them that are differentially expressed over development and in a particular cell type. Then we can look for enrichment within those genes of we can look for whether there's an overrepresentation of genes that are differentially expressed in a certain cell type with a certain trajectory over development for that particular disorder. So essentially what this is trying to do is to associate the particular cell type and the expression dynamics of disease-associated genes. So to some degree, this may indicate when and where dysfunction in the normal expression of that gene might be relevant to that disease. As an example, we notice this group of disorders, which are multiple cognitive disorders that occur in later life, dementia, mild cognitive disorder, amnesia, Huntington's disease. What we saw is that, in particular, they showed an enrichment of genes that are transiently upregulated. So they're upregulated, then downregulated, and they're upregulated postnatally in particular principal neuron and inhibitory neuron subtypes, and they're highly enriched for having roles in synaptic signaling. So we think this could indicate that this is a period of critical formation of these complex neuronal circuitry and synaptic connections and activity that later on is sensitive to being disrupted in these cognitive disorders in late life. And of course, for any of these disorders, since we have groups of genes that are differentially expressed, we can focus on particular cell types and developmental dynamics of genes that are associated with a particular disorder and predict transcription factors that might be that are enriched in those and that might be linked to that could be disrupted, which could contribute to dysregulation of those genes that are associated with that disease. For example, phosphagen frequently showing up associated with impaired cognition and linked to memory mechanisms. I'll skip over this example in the interest of time. So the final thing we want to do is to use this reference map of cortical development to map other samples to it, to try to be able to then further analyze single nucleus RNA-seq data sets from other healthy samples or disease samples, or indeed the organoid models that we were working on originally. So we developed a stable integration process that allows you to take a single nucleus RNA-seq data set and integrate it into our map reliably. What we could do is take a range of different prefrontal cortex single nucleus RNA-seq samples that have been produced by an independent study recently, and map them into our reference and predict through nearest neighbor analysis what the age of the nuclei in those samples were. And so here we're plotting the distribution of the ages of the nuclei that we identify from each sample. And we can see that they broadly correlate with the known age of these samples, whether they're fetal or newborn, through to 1-year-old, through to aging through development. We can use this to integrate disease samples as we do for glioblastoma and identify the cell types and which particular cell states are present in the disorders. But what we want to focus on predominantly is this organoid maturation. So in the last couple of minutes, I'll just focus on this. So we had been growing these organoids before and seen broadly the lack of maturation based on DNA methylation states. What we want to do is further study this now at the single cell level and see, is this happening similarly to all cells within the organoid? So we generated these cerebral organoids from human ES cells that had contained the cell types that we expect to have, that showed indeed some decrease in markers of immaturity and increase of markers in maturity as we age the cerebral organoids from 3 to 9 months. And we did single nucleus RNA-seq with the help of Luciano Martelotto at different stages through this development, 5 of different ages of culture of the organoids, 5, 9, or 12 months. And we integrated the organoid nuclei into our reference map, which you can see by the colored dots here. Indeed, what we saw was that only in older organoids, 9 or 12 months, did we begin to see some nuclei from the organoids that mapped to postnatal stages in the real human brain. It takes a long time, and we only get about 15% of the neurons that would show this maturity. We could then do a couple of comparisons. What are the differentially expressed genes between the postnatally mature organoid neurons versus the immature organoid principal neurons? And indeed, what we see is not surprising. It makes sense that we see a range of genes downregulated in the organoid postnatal neurons that are associated with early differentiation and development, and a range of genes that are upregulated in the postnatal neurons that are characteristic of neuronal maturation, ion transmitters, ion channels, neurotransmitters, and many genes that are normally upregulated. So this makes sense. But then we could take the organoid neurons that align to postnatal stages in the real brain, and we could say, well, okay, are there any difference between those postnatally mature organoid neurons and their nearest neighbor brain neurons in the real brain samples? What we see is that even though they show characteristics of being postnatally mature, we see extensive differential expression still in these organoid mature neurons. We see broad downregulation of a range of genes in the postnatal neurons in the organoids, and these are associated with synapse organization, structure, activity, and regulation, so key neuronal processes. We can then take these genes that are downregulated in the postnatal neurons, and through a network analysis, predict transcription factors that may be regulating these gene expression differences. And what we see is this identifies a range of transcription factors that are normally upregulated in normal brain development, that are downregulated in the organoid postnatal neurons, and it suggests that the function of these may be lacking. The upregulation, the activity of these regulators may be lacking within the organoid neurons. But it suggests that the neurons we're creating may lack some of the proper synaptic connectivity networks. But these are the type of transcription factors that we want to now take, and we are now taking through screens to see if we can activate them in isolation or combination in the organoids to force the maturation and further maturation. Okay, and I'm going over time here, so I'll just wrap up, but hopefully what I've shown you here is a range of different features that we can pull out of this data set, and hopefully what we've generated is a useful resource for further targeted analyses into how the transcriptional dynamics or regulators over development in many different cell types, as well as useful tools to try to, and information to try to progress these in vitro models and advance their utility for disease modeling or for mechanistic studies. To just finish here, acknowledging the people who drove this. So in particular, this is co-led by four researchers in my group: Chuck Herring, Becca Simmons, Saskia Freitag, and Daniel Popper, as well as a range of different collaborators in my lab, as well as collaborators from UNSW, Perkins and UWA, University of Adelaide, and Spain. Particularly like to thank the NIH Neurobiobank, which is a really unique and generous resource and bank as well as the donor families who donated these samples at what must have been the most difficult point in their life. And with that, I'll just say that I have multiple postdoc positions that are opening in my lab if this type of work is of any interest. And thanks for listening.